Good evening to all. I hope I am audible to you. Good evening to all. I hope I am audible to you. Good evening to all. I hope I am audible to you. Good evening to all. I hope I am audible to you. Good evening to all. I hope I am audible to you. Good evening to all. I hope I am audible to you. Good evening to all. I hope I am audible to you. Good evening to all. I hope I am audible to you. Good evening to all. I hope I am audible to you. Good evening to all. I hope I am audible to you. So today we'll see the remaining points. So what we have seen yesterday we have seen the mutar rotation. Okay, we have seen the mutar rotation, that is a change in the optical rotation due to interconversion of that alpha D glucose and beta D glucose, that is known as the mutar rotation. And we have already seen the anomers and epimers. What do you mean by anomerism? What do you mean by epimerism? Right. So this is a alpha D glucose. Okay. If you check the optical rotation of freshly prepared glucose solution, dextrose solution, it is around plus 112 means dextro. Okay. Then this alpha D glucose changes, the optical rotation changes to this 52.5, which is intermediate, and lastly gets stabilized at a 19. Stabilized at a 19. That is a due to the beta isomer. Right. So alpha D glucose has a optical rotation of 112 and beta D glucose has optical rotation of plus 19. That means interconversion of optical rotation due to the change in the optical rotation due to the interconversion of alpha and beta isomers to get an equilibrium, that is known as a mutar rotation. Okay. So for the fructose, initially optical rotation of initial solution of fructose is a minus 21, means LEVO, right? That changes to a minus 22 and lastly get stabilized at a minus 133.5, right? Beta isomer, alpha isomer of fructose is stable, beta isomer of glucose is stable. So we have seen the epimers. Remember, glucose and mannose, they are known as the C2 epimers. Mannose and glucose. Okay, so mannose, D-mannose differs in the arrangement of EH and OH at the carbon number 2. That's why mannose is a C2 epimer of glucose, and then galactose differs in the structure at a carbon number 4. That's why galactose is a C4 epimer of glucose. So glucose and galactose, they are the C4 epimer. Glucose and mannose, they are the C2 epimers. Okay, we have already seen. Right? We have seen this is a Fisher's projection formula. So linear after cyclation, Fisher projection formula, and hello, कोण? बोला.
सर नाही पेट एक्झाम
मी 8 वाजता फोन करू, मी लेक्चर मध्ये आता 8:15 वाजता. We have seen about the furanose and pyranose structure. So furanose structure, so if glucose molecule is cyclized and if you draw a structure like this, this is known as a Fisher formula, and this structure will look like a pyran. That's why pyranose. This is known as a Haworth formula. Okay. Fructose So cyclic structure of fructose will look like a furan. That's why furan was. We have seen the physical properties. So iodo compounds, that aldose sugar when treated with the hydroiodic acid, they form a iodo compounds. One of the physical property, acetylation or ester formation. So when treated with the acetyl chloride, sugars when treated with the acetyl chloride to form a ester due to presence of alcoholic OH groups. They also react with the other acids to form a corresponding esters. We have seen the oxazone formation. So what is the reagent of oxazone? उसके ऊपर क्वेश्चन आ चुका है already. The reagent in the oxazone test is nothing but the phenyl. Remember, phenyl is a reagent of oxazone formation. And remember, only carbon number 1 and carbon number 2 of the sugar reacts with the phenyl to give a respective hydrogens. These hydrogens, they have specific crystal and shape. Based upon that, they can be distinguished. That's why oxazone formation is the test to differentiate glucose, fructose and monosaccharides, disaccharides. Okay, देखो, oxazone formation is also one of the tests to distinguish between these monosaccharides and disaccharides, because this glucose, mannose and fructose, they will give you same color, same crystal known as a glucosazone, that is needle shaped crystal or needle shaped bundle of hay. Lactosazone is a powder puff, maltosazone is a sunflower shape. Okay, we have seen this. Okay, so this is known as this structure, or this is a needle shaped hay structure given by glucose, fructose and mannose. This glucose, fructose, mannose, when treated with the phenyl, they will give you glucosazones. Then lactose will give you powder puff shaped crystal, and maltose will give you sunflower shaped crystal. ये तीनों के ऊपर ऑलरेडी क्वेश्चंस आ चुके हैं. You can say 3 times question has been there. There is one test in pharmacognosy also. You might have studied. When starch is treated with the iodine solution, starch will give you blue color with the iodine solution. Starch hydrolyzed, okay. When starch is hydrolyzed, so it will become a soluble starch. Soluble starch will also give you blue color with the iodine. Then soluble starch, if hydrolyzed, you will get a 2-part, amylose and amylodextrin. So amylodextrin will give you purple color with the iodine. Then erythrodextrin will give you red color. Then acrodextrin will give colorless. Okay? Then will give maltose. So remember here also, so this is aldose sugar. So it may be either glucose, fructose, mannose, glucose, fructose, mannose. So that react with the phenyl. So first carbon atom of पहला carbon atom is functional carbon that reacts with the phenyl to form intermediate, that is phenyl. Then again second carbon react with the phenyl, then intermediate, okay? And finally the first 2 carbon atom reacts with the phenyl to form a respective phenyl or oxazones. These are the structure of glucosazone, glucose, maltose, glucose, then mannose, glucose, mannose and fructose. They will all give you needle shaped crystals, glucose, mannose, and then maltose will give you sunflower shaped crystal. Lactose will give you powder puff shaped crystal or cotton ball shaped crystal. ये crystal के ऊपर क्वेश्चन आ चुका है ऑलरेडी 2-3 बार, right? Now interconversion of sugar, we have seen that in a weakly basic medium, glucose, mannose, fructose, they undergo conversion and formation of common inert oil, which forms from which tautomerases to a 3 sugar. मतलब interconversion of glucose, mannose and fructose, glucose, mannose, fructose, they gets interconverted into each other via formation of common inert oil, that is known as a Lobre-Deverne-Van Eckestein reaction. Lobre the burn ecostine van ecostine reaction. Then oxidation product, इसके ऊपर क्वेश्चन आ चुका है. So depending on the oxidizing agent and the depending on the oxidizing agent and carbon involved, carbon oxidizer, so they are oxidized to aldonic acid, then aldonic acid means glucose when treated with the bromine will give you gluconic acid or aldonic acid. Then saccharic or alderic acid, so oxidation of D glucose with the nitric acid. ना अगर ब्रोमीन के साथ में रिएक्ट होगा तो ग्लूकोनिक एसिड देगा, नाइट्रिक एसिड के साथ में अल्डेरिक और सैकरिक एसिड देगा. एंड इफ टर्मिनल अल्कोहल ग्रुप इज ऑक्सीडाइज्ड so that will give you uronic acid. Means if glucose is oxidized and if terminal alcohol group is oxidized, so that will give you glucouronic acid. Glucouronic acid is very important in body. Reduction means है ना, so sugars are reduced by sodium amalgam to respective alcohol. जो शुगर है, शुगर रिडक्शन होके अल्कोहल बनेगा, है ना. तो ग्लूकोज का रिडक्शन होके sorbitol बनेगा. Galactose after reduction that will give you dulcitol. Mannose will give you mannitol. Fructose will give you mannitol and disorbitol. We have seen yesterday. You can see it is a structure of glucose. Structure of glucose will form a inert oil in the basic medium. Basic medium में क्या होगा, इसका inert oil होगा. This shifting of this, है ना, shifting of this double bond to here, यहाँ पे डबल बॉन्ड आ जाएगा, यहाँ पे हाइड्रोजन आ जाएगा एंड डबल बॉन्ड यहाँ पे आ जाएगा, तो यहाँ का हाइड्रोजन चला जाएगा. तो शिफ्टिंग ऑफ हाइड्रोजन, है ना, to form a inert oil, and from this inert oil you can get a mannose, from this inert oil you can get a fructose. So interconversion of these fructose, mannose and glucose via formation of a common inert oil known as a mutarotation, known as a enolization, known as enolization. So glucose when oxidized, if the functional group, that is if aldehyde group is oxidized, ना अगर aldehyde group oxidize होता है ग्लूकोज का, you will get a gluconic acid, you will get a gluconic acid, मतलब ब्रोमीन के साथ में रिएक्शन करके ग्लूकोनिक एसिड. ठीक है. अगर नाइट्रिक एसिड के साथ में if glucose is oxidized with the nitric acid, then there is a oxidation of both, you know, aldehyde group also and terminal alcohol group also, that leads to glucosaccharic acid. And if glucose is oxidized with the if terminal alcohol group of alcohol only gets oxidized, you will get a glucouronic acid ना. मतलब अगर फंक्शनल ग्रुप ऑक्सीडाइज होता है शुगर का, तो दैट विल गिव यू gluconic acid. If terminal alcohol group is only oxidized, you will get a glucouronic acid. And if both are oxidized, then you will get a glucosaccharic acid. Reduction of D glucose will give you D-sorbitol. यहाँ पे रिडक्शन मतलब क्या है, एडिशन ऑफ, है ना, हाइड्रोजन CH2 that leads to corresponding alcohol sorbitol. Yesterday we have seen this tautomerization or enolization. Tautomerization or enolization we have seen, that is a shifting of hydrogen bond, from one carbon to another carbon to produce a common inert oil, known as a tautomerization. So tautomerization or enolization, है ना, tautomerization को ही बोलते हैं अपन enolization. What do you mean by tautomerization or enolization? Shifting of hydrogen bond from one carbon to another to produce an inert oil, known as a tautomerization. Enolization. So glucose, fructose, mannose we have seen will form a common inert oil from which each can be interconverted. Oxidation, colamine, depending on the oxidizing agent, whether aldehyde or terminal alcohol group is oxidized or both are oxidized, depending on that we'll get a gluconic acid or glucouronic acid or glucosaccharic acid. मतलब if aldehyde group is oxidized, you will get a gluconic acid. If terminal alcohol group is oxidized, you will get a glucouronic acid. And if both are oxidized, you will get a glucosaccharic acid. reduction है ना, reduction of sugar will give you corresponding alcohol. Glucose will give you sorbitol, mannose will give you mannitol. Oxazone formation, only first two carbons are involved in the oxazone formation. We have seen that. So, you know, treatment with the acid. Pentose sugar, when treated with the acid, you will get a furfural aldehyde derivative. Furfural. You know. Then again, you can see sugar with the acid. When sugars are treated with the acid, hydrolysis of polysaccharide with the mineral acids 0.5 to 1 normal causes the decomposition of monosaccharide. Pentose gives cyclic aldehydes known as a furfural aldehyde. Furfural. The product combines with the phenol to give color reaction. तो क्या है कि when sugars are treated with the acid, so there is a decomposition of acid, and for example pentose gives cyclic aldehyde, furfural aldehyde. Now this furfural aldehyde will combine with the phenol to show the color reaction. It is the basis of identification test. So hexose sugar will give hydroxymethyl furfural. मतलब अगर pentose sugar है तो give you furfural, and the hexose sugar is there, it will give you hydroxymethyl furfural that can react with the phenol, जैसे alpha naphthol. There is one test known as a Molish test. जो आइडेंटिफिकेशन टेस्ट होता है शुगर्स का, carbohydrates are identified by few tests. One of the tests is the Molish test. So what is the mechanism in a Molish test? So sugar are treated with the acid, then there is a production of furfural or either hydroxymethyl furfural. So that react with the phenyl to give color reaction. Now monosaccharides with the alkali. If monosaccharides are treated with the dilute alkali, that leads to mutar rotation. अगर डाइल्यूट अल्कली के साथ में डाइल्यूट बेसिक सॉल्यूशन के साथ में अगर मोनोसैकेराइड का रिएक्शन क्या होगा, मोनोसैकेराइड रिएक्ट विद द डाइल्यूट बेसिक सॉल्यूशन एंड देयर इज अ रिजल्ट इन द म्यूटर रोटेशन. देयर इज कन्वर्जन ऑफ अल्फा टू द बीटा, ऑफ अल्फा डी sugar to the beta D sugar. Now if monosaccharides are treated with the concentrated alkali solution or concentrated basic solution, there is a caramelization. Of sugar. That is nothing but the decomposition of sugar which gives you yellow or brown pigment known as a caramelization. Question already आ चुका है कि when sugars are treated with the acid, that leads to formation of furfural aldehyde or hydroxymethyl furfural. But in a basic solution, in weak basic solution mutar rotation, in strong basic solution caramelization. ठीक है, याद रखना है, सिंपल है. देखो, when monosaccharides are treated with the acid के साथ में ट्रीटमेंट होगा तो क्या होगा, that will give you furfural. If it is a pentose sugar, that will give you furfural, and if it is hexose sugar, that will give you hydroxymethyl furfural. So this hydroxymethyl furfural combines with the alpha naphthol or phenol to give a color reaction, which is the basis for identification of. Again, monosaccharides when treated with the dilute alkali leads to mutar rotation. When treated with the concentrated alkali, that gives, when treated with the strong basic solution, gives caramelization of sugar. है ना? Now reducing action of sugar in the alkaline solution. Reducing action of sugar in the alkaline solution. मतलब if sugars है ना, sugar solution है ना, in alkaline medium, जो sugar है in alkaline sugar containing free aldehyde or free ketone group है ना, in alkaline solution sugar containing free aldehyde or ketone group undergo enolization. The enediol forms sugar, enediol formed of a sugar, they are very reactive and they get easily oxidized by oxygen and other oxidizing agent. मतलब जो मोनोसेक्राइड से, जो शुगर से ना, अल्कलाइन सॉल्यूशन में क्या होगा उसका, है ना, अल्डिहाइड बनेगा and undergo enolization. Enodiols, they are reactive, and they get easily oxidized by oxidizing agent like silver, है ना, mercury, है ना, copper, etc. to form a sugar acid, and this is the test. This is a basis of identification of reducing and non-reducing sugar. It is the basis of identification of whether sugar is a reducing sugar or non-reducing sugar. एक बार फिर से देखो, reducing action of sugar in the alkaline solution. In an alkaline solution, sugar containing free aldehyde or ketone group that undergo enolization. The enediol formed, they are reactive and oxidized by oxygen or other oxidizing agent like silver, mercury, copper, to form a sugar acids. Based on this, you can see sugar in a basic medium, enediol reduces the copper, ये है copper, Cu plus Cu double plus cuprosine from the copper sulfate. Copper sulfate is a reagent of Benedict. बेनेडिक्ट reagent है, जो बारफर reagent है, उसमें copper sulfate होता है. So enediol, they are reactive, they get oxidized due to the cuprosine. Cuprosine is converted into cupric ion, U plus cupric ion. Now cupric ion, है ना, combine with the hydroxide to make cuprous oxide, which is having red color. So this is the basis of Benedict reagent. एक टेस्ट है आपका बेनेडिक्ट टेस्ट. दिस टेस्ट इज यूज्ड टू डिस्टिंग्विश बिटवीन रिड्यूसिंग शुगर एंड नॉन रिड्यूसिंग शुगर. जो शुगर्स होते हैं अल्कलाइन सॉल्यूशन में इनाडायोल फॉर्म करते हैं, राइट, एंड देन इट विल बिकम अ शुगर एसिड. बट दिस इनाडायोल्स, दे आर reactive, they reduces the ions, for example cuprous ion into cupric ion, and cuprous ion has a different color and cupric ion has a different color. So this reaction, color reaction, can be used to identify the whether sugar is a reducing sugar or non reducing sugar. मैंने बोला है कि when sugars are treated with the acid, the pentose sugar है, when treated with the acid, you will get a furfural. And this furfural react with the phenol or alpha naphthol to give a color reaction, which is a basis of Mollish test. पहला जो टेस्ट होता है Mollish test, उसका बेसिस क्या है, sugars treated with the acid form a furfural, है ना, that furfural will react with the phenol or naphthol to give a color reaction, है ना. अगर पेंटो शुगर है तो दैट विल गिव यू फुरफुराल. इफ इट इज अ हेक्सो शुगर, दैट विल गिव यू हाइड्रोक्सीमिथाइल. अदर इंपॉर्टेंट शुगर डेरिवेटिव, और बाकी शुगर्स के डेरिवेटिव कौन से हैं, तो अदर इंपॉर्टेंट डेरिवेटिव ऑफ शुगर, तो देयर इज अ डीऑक्सी शुगर. व्हाट डू यू मीन बाय डीऑक्सी शुगर? एज द नेम इंडिकेट, डीऑक्सी, तो रिमूवल ऑफ ऑक्सीजन. As the name indicates, deoxy means removal of oxygen. So sugar in which oxygen of hydroxyl group has been removed. So deoxy sugar मतलब क्या है? Sugar from which the oxygen and oxygen of OH group is removed. Sugars from which oxygen of OH group has been removed, leaving the hydrogen. So that's why if the CHOH is there, that is converted into CH2, and if CH2OH is there, then that is converted into CH3. So that is a deoxy sugar. So 2-deoxy D-ribose, this sugar is found in the DNA, and 6-deoxy L-galactose, this sugar is found in the glycoprotein. तो deoxy sugar मतलब क्या है? Removal of oxygen from the OH group leads to deoxy sugar. Another type of sugar is amino sugar. Amino sugar है ना, sugar derivative है. Deoxy sugar is a sugar derivative. Amino sugar is a sugar derivative है ना. Glycoside is also sugar derivative. Amino sugar acids are also derivative of sugar है ना. So sugar with the amino group known as amino sugar. There are two type of amino sugar ना. ग्लाइकोसामाइन है ना, पहला कौन सा है? ग्लाइकोसाइलामाइन and second is a glycosamine. As the name indicate, amino sugar means what? There is a amino group in the sugar. As the name indicate, amino sugar means there is a amino group in the sugar. So they are, for example, a hexosamine. Hexo sugar with the amine known as a hexosamine. Amino sugars are hexosamine. Sugar with the amino group. There are two type of amino sugar. पहला कौन सा है? Glycosylamine. So in a glycosylamine, anomeric OH is replaced with the amino. Means functional group OH जो है आपका, that is replaced with the amino group. Example is a ribosylamine, which is involved in the purine synthesis. Second type of amino sugar is a glycosamine. So alcoholic OH जब replace होता है, when alcoholic OH is replaced by NH2, so that leads to glycosamine. Glycosamine का example क्या है? Example is a glucosamine. So it is a component of chitosamine, because जो microbial cell wall होता है उसमें chitin होता है, and chitin is made from a polymer of N-acetyl glucosamine. ना ग्लाइकोसामाइन का एग्जांपल है ग्लूकोसामाइन. एंड ग्लूकोसामाइन क्या है? इट इज अ पार्ट ऑफ माइक्रोबियल सेल, दैट इज अ पार्ट ऑफ माइक्रोबियल सेल वॉल, दैट इज अ चिटिन. एंड चिटिन इज अ पॉलीमर ऑफ N-acetyl glycosamine, N-acetyl glucosamine. That's why glucosamine is also known as a chitosamine. गैलेक्टोसामाइन मतलब गैलेक्टोज है, यहाँ पे अमाइनो है. गैलेक्टोसामाइन है ना, it is found in the chondroitin sulfate, which is a part of bones and cartilage. That's why it is also known as chondrosamine. ग्लूकोसामाइन को क्या बोलते हैं? चिटोसामाइन. क्यों? बिकॉज ग्लूकोसामाइन कहाँ पे होता है? ग्लूकोसामाइन इज प्रेजेंट इन द चिटिन. चिटिन इज अ पार्ट ऑफ माइक्रोबियल सेल वॉल, which is a polymer of N-acetyl glucosamine. That's why glucosamine sometimes also called as chitosamine. Galactosamine also sometimes called as chondrosamine. Why? Because galactosamine is found in the chondroitin sulfate, which is a part of bones and cartilage. Amino sugar acid. तो है न्यूरामिनिक एसिड and muramic acid. Neurominic acid and muramic acid. Neuramic acid is a aldol condensation product of pyruvic acid and D-menosamine. है ना, ये अमाइनो शुगर है, एंड अमाइनो शुगर से बनता है अमाइनो शुगर एसिड्स. न्यूरामिनिक एसिड विच इज अ कंडेंसेशन, aldol condensation product of pyruvic acid and D-menosamine. जो मैनोज है उसको अमाइनो ग्रुप लगा दिया तो मेनोसामाइन, मैनोज को अमाइनो ग्रुप लगा दिया तो मेनोसामाइन बन जाएगा, और उसका और पायरुविक एसिड का कंडेंसेशन करेंगे तो वो बनेगा आपका न्यूरामिनिक एसिड. अनस्टेबल एंड फाउंड इन द नेचर इन द फॉर्म ऑफ, है ना, एसिलेटेड डेरिवेटिव. मतलब न्यूरामिनिक एसिड इज अनस्टेबल एंड दैट्स व्हाई इट इज फाउंड इन द नेचर एज एसिलेटेड डेरिवेटिव. एसिलेटेड डेरिवेटिव ऑफ सियालिक एसिड नोन एज नाना मतलब N-acetyl न्यूरामिनिक एसिड. नाना मतलब क्या होता है? N-acetyl न्यूरामिनिक एसिड which is a component of mucopolysaccharide. न्यूरामिनिक एसिड है ना, म्यूरामिक एसिड. म्यूरामिक एसिड is a condensation product of lactic acid with the D monosamine and it is present in the bacterial cell wall. Bacterial cell wall में ये भी होता है uramic acid. Now glycosides मतलब क्या? So when a glycone part, it may be either phenol or it may be either alkaloid. When it combine with the glycol or sugar by a glycosidic bond, that is a glycoside. Okay? Glycoside मतलब क्या? A non-sugar portion when bind to the sugar portion via glycosidic bond to form a glycoside. It is found in the plants and antibiotic. जो कुछ एंटीबायोटिक है, ग्लाइकोसाइड एंटीबायोटिक, ग्लाइकोपेप्टाइड एंटीबायोटिक्स, या फिर जो आपके ग्लाइकोसाइड से इसमें भी ये ग्लाइकोसाइड बॉन्ड होता है. You can see structure of D-glucosamine. D-glucosamine मतलब क्या? ये D-glucose का स्ट्रक्चर है. It is a structure of D-glucose. Just at a carbon number 2, OH is replaced by amino group. OH is replaced by amino group to form a D-glucosamine. This is a structure of galactose and just second OH of galactose is replaced by amino known as D-galactosamine. Right? To estimate the pentose sugar in the carbohydrate, if you have carbohydrate, to estimate the pentose sugar, whether there is a pentose sugar or not in a carbohydrate, furfural decomposition product of pentose sugar combined with the fluoroglycinol, the complex of both is used to estimate the pentose. तो पहला जो होता है मॉलिश टेस्ट, alcoholic solution of alpha naphthol, the reagent, the molish reagent. Molish test जो होता है, that is nothing but the alcoholic solution of alpha naphthol. Alpha naphthol क्या है? Phenol. Sugar gives a red violet ring. है ना? क्यों? बिकॉज इसमें क्या है कि है ना, शुगर का सोल्यूशन में अल्फा नेप्थोल डालने का है आपको, अल्कोहलिक सोल्यूशन अल्फा नेप्थोल, एंड यू हैव टू ऐड acid by side wise. So what happen? So in the acid, the sugar is converted into furfural and that react with the phenol to give a color. This test is the non-specific test given positive by all sugar. So identification test for sugar is a Molish test. इसका basis क्या है? What is the basis of Molish test? Basis of Molish test is nothing but the when sugars are treated with the acid, they form a furfural or hydroxymethyl furfural, and that hydroxymethyl furfural or furfural reacts with the phenol like alpha naphthol to give a color reaction given positive by all tests. And Selivanov test is there. Selivanov test, so sugar known as a keto sugar will form a cherry red color with the resveratrol, है ना. keto sugar अगर है जिसमें कीटोन फंक्शन है, तो कीटोन react with the resveratrol to form a cherry red color. It is a specific test for D fructose. Question already ऊपर आ चुका है. How you can distinguish between glucose and fructose? There is a two test, Selivanov test, rapid furfural test, and there is one more test. Basically there are three tests to distinguish between aldoses and ketoses. देखेंगे अपन ये बाद में. Now failing solution. Failing solution क्या है? Sodium potassium tartrate. It is a solution of Roche salt. Roche salt in the sodium or potassium hydroxide and copper sulfate. It will form in a diol formed in a basic medium. Copper is converted into cuprous oxide, red PPT. It is a fillings test. We'll see it. Right. So different chemical test for carbohydrate. Very important question, multiple times आ चुका है. Chemical test for identification of carbohydrates. You might have performed a practicals in your college. First test is nothing but the Molish test. It is a test for identification test for identification or detection of carbohydrates. इसमें क्या होता है, sugars are hydrolyzed with the sulfuric acid and then treated with the alpha naphthol. First of all, sugar solution is hydrolyzed with the mineral acid and then alpha naphthol is added. We get a violet colored complex. That means there is a presence of sugar. Polish test. Second test is very important question भी आ चुका है इसके ऊपर. Benedict test. Benedict test. What is Benedict test? It is a specific test to distinguish between reducing sugar and non-reducing sugar. Multiple times question आ चुका है. How you can distinguish between reducing sugar and non-reducing sugar. Remember Benedict test. Benedict test is a test to distinguish between reducing and non-reducing sugar. What happens in this Benedict test? क्या होता है इसमें? So sugars under alkaline condition, they forms inert oils. Inert oils, they are reactive. They reduces the cupric ion. They reduces the cupric ion Fe2+ to the cuprous ion Fe. Cupric ion has a different color, cuprous ion has a different color, which forms yellow precipitate, which forms a yellow precipitate of cuprous hydroxide to red precipitate of cupric oxide. So if you get a red precipitate of cupric oxide, that means there is a reducing sugar. Reducing sugar मतलब क्या है? Sugar having either free aldehyde or ketone group. जो भी शुगर में फ्री aldehyde ग्रुप होगा, ketone ग्रुप होगा, that is known as a reducing sugar, right? कि फ्री aldehyde ketone ग्रुप है तभी जाके क्या बनेगा, inert oils बनेंगे. If there is a free aldehyde or ketone, then it will form inert oils in the basic solution. That inert oil can reduce the oxidizing agent like copper and all that. This was a question multiple times asked in GPAT Niper DI Pharmacist. Again, another test to distinguish between reducing sugar and non-reducing sugar is a Barford test. Second test to distinguish between reducing and non-reducing sugar. So this test is again same as that of the Benedict test, only this test is carried out in the acidic medium, strong acid. What is the difference between Benedict test and Barford test? Both tests are used to distinguish the reducing and non-reducing sugar, but only Benedict test is carried out in the alkaline medium and Barford test is carried out in the acidic medium. So testing and Barford test is used to distinguish between the monosaccharides and disaccharides. Now Selivanov test we have already seen. Cherry-red complex will be formed with the HCl and resorcinol. Is a specific test for ketoexosis. It is a specific test for fructose or ketoexosis. Now Fulger test. Fulger test is again specific test for ketoexosis. The blue color that occurs with the stannous chloride and urea. Rapid furfural test. Purple color complex with the hydrochloric acid and alpha naphthol. Then specific test for ketoexosis. मतलब देखो यहाँ पे Molly's test is given positive by all sugars. Benedict test given positive by only reducing sugars. Barford test given positive by monosaccharides. Selivanov test is given positive by fructose or ketoexosis. Selivanov test, Fulger test and rapid furfural test. These three tests are tests to distinguish between aldose and ketose sugar. If there is a ketose sugar, ketone function group वाला sugar है, तो Selivanov test, Fulger test and rapid furfural test, all these tests will be positive. Now Osazone formation test. Osazone test. So we have seen that phenyldrazine is used as a reagent. Glucose, fructose, mannose, they will give you same needle shaped crystals. Maltose will give you sunflower shaped. lactose will give you powder puff shaped crystals. जो ना बच्चों को नहीं कि आप पाउडर लगाते हैं पाउडर पफ की शेप की जैसे क्रिस्टल, राइट. So this question was also multiple times. What type of crystals are given by maltose? Maltose will give you sunflower shaped crystal, lactose will give you powder puff shaped crystal, and glucose, fructose, mannose, they will give you needle shaped crystal. Another test is there, sucrose hydrolysis test. Sucrose क्या है? What is sucrose? Sucrose is a disaccharide. Sucrose is a disaccharide and sucrose is a non-reducing sugar. Sucrose क्या है? Non-reducing sugar. Example याद रखो, क्वेश्चन आता है. Sucrose is a non-reducing sugar. Why? Because in a sucrose, glucose and fructose are combined, and in a 1-2 linkage. मतलब one first carbon atom of your glucose is a aldehyde containing carbon, and second carbon of fructose, that is a ketone containing carbon. Both are involved in the glycoside bond formation. That's why there is no free aldehyde or ketone group in the sucrose, and that's why sucrose is a non-reducing sugar. You know, sucrose will not give you Benedict test and Barford test. Benedict test, Barford test जो है, sucrose will not give you positive because sucrose में there is no aldehyde and ketone group which is a free, right? So therefore sucrose is first hydrolyzed with the acid, and then environment made is alkaline, and then it will give you Barford and Benedict test positive. Iodine test. Iodine test क्या है? पॉलीसेकेराइड के लिए होता है. पॉलीसेकेराइड फॉर्म्स अ कलर्ड कॉम्प्लेक्स विद आयोडीन. स्टार्च विल गिव यू ब्लू कलर. इसके ऊपर मल्टीपल टाइम्स क्वेश्चन आ चुका है. स्टार्च विल गिव यू ब्लू कलर. स्टार्च विल गिव यू ब्लू कलर. Dextrans will give you red color. Now starch जो है, it is a polysaccharide which is composed of two compounds. Starch जो होता है, that is a polysaccharide composed of amylose and amylopectin. Now amylose with iodine will give you blue or black color. Amylopectin will give you reddish brown color. इसके ऊपर भी क्वेश्चन आ चुका है GPAT 2019 में. Remember, starch is a polysaccharide of glucose and fructose. So glucose so starch is having two components. Starch में दो component होता है. First one is amylose and second one is amylopectin. Amylose will give you blue or black color. Amylopectin will give you reddish brown color with the iodine. Was a question in GPAT 2. ये तो मल्टीपल टाइम्स आ चुका है. ये भी मल्टीपल टाइम्स आ चुका है. Whatever I am explaining you, so there is a question on this part. Now ना, जैसे मैंने बोला है, I already told you that Benedict test is very important, and they have already asked questions on Benedict test. So what is the use of Benedict test? Benedict test is used to distinguish between reducing sugar and non-reducing sugar. Now what is the basis of that test? Basis of Benedict test, again I will explain. Sugar plus alkali, what will sugar plus weak alkali solution, there is a formation of enediols, common enediols. Enediols जो होता है, they are reactive, they are reducing agent, they reduces the cupric ion to the cuprous ion, Cu double plus to the Cu plus reduced. और ये cuprous ion कहाँ से आता है? Benedict reagent होता है. What is the reagent of Benedict? तो Benedict reagent होता है ना, कॉपर सल्फेट, है ना, कॉपर सल्फेट या फिर कॉपर कॉम्प्लेक्स कॉपर टार्ट्रेट एंड साइट्रेट. So from that cupric ion is reduced to the cuprous ion, right? Now this cuprous ion reacts with the OH hydroxide ion to form a cuprous hydroxide which is having yellow color, and on heating this cuprous hydroxide gets converted into cuprous oxide which is having red color. And this is a red color means positive test. This indicate the test positive means what? If a sugar has a free aldehyde or ketone group, अगर शुगर में फ्री aldehyde है या फिर कीटोन ग्रुप है ना, मतलब फ्री मतलब क्या, दैट इज नॉट इन्वॉल्व इन द बॉन्ड. So if sugar has a free aldehyde or ketone group, so Benedict test will be positive. Remember, what do you mean by reducing sugar? The sugar which is having free aldehyde group known as a reducing sugar. The glucose में free aldehyde group है, fructose में free ketone group है ना, maltose में, lactose में free aldehyde group है, but sucrose में है क्या, नहीं है. So that's why sucrose is a non-reducing sugar. Sucrose is a non. Now we have seen the monosaccharide. Now disaccharide means they are having 2 units. So basically the term oligosaccharide, oligo means few, oligo मतलब क्या होता है, oligo means few monomers. So 2 to 10 monomers are present in the oligosaccharides. They are also sweet in taste. Oligosaccharides also are divided into 2 types. Disaccharides means 2 monomers and trisaccharide means 3 monomers. Tetra means 4, like that. Disaccharides means 2 monosaccharides are joined by glycosidic bond. पहला है आपका सुक्रोज. सुक्रोज क्या है, इज अ डाइसैकेराइड. सुक्रोज इज आल्सो नोन एज अ, सुक्रोज को क्या बोलते हैं अपन, सुक्रोज इज नोन एज अ केन शुगर और टेबल शुगर और कॉमन शुगर, टेबल शुगर और कॉमन शुगर और केन शुगर. Now sucrose is a disaccharide which is made from one molecule of glucose and one molecule of fructose, है ना. मतलब सुक्रोज जो है आपका, जो टेबल शुगर है, केन शुगर है ना, उसमें क्या है, ग्लूकोज मॉलिक्यूल है, 1 ग्लूकोज मॉलिक्यूल 1 फ्रुक्टोज मॉलिक्यूल, है ना. ग्लूकोज प्लस फ्रुक्टोज विल गेट अ फ्रुक्टोज. नाउ ग्लूकोज इज लिंक्ड विद द फ्रुक्टोज. लिंकेज का है अल्फा डी ग्लूकोसिल, मतलब अल्फा डी ग्लूकोज 1,2, मतलब फर्स्ट कार्बोनेट ऑफ ग्लूकोज एंड सेकंड कार्बोनेट ऑफ बीटा डी फ्रुक्टोज ना. तो अल्फा डी ग्लूकोसिल 1,2 बीटा डी फ्रुक्टोज, ये इसके ऊपर क्वेश्चन आ चुका है. व्हाट इज द बॉन्ड, ग्लाइकोसिडिक बॉन्ड लिंकेज, व्हाट इज द ग्लाइकोसिडिक बॉन्ड लिंकेज. लिंकेज क्या है फिर, अल्फा डी ग्लूकोसिल 1,2 बीटा डी फ्रुक्टोज इज अ सुक्रोज. Sucrose is a non-reducing sugar. Remember, made from a glucose plus fructose. Sucrose is also known as a cane sugar, also known as a table sugar, also known as a common sugar, also known as a invert sugar. Invert sugar क्यों बोलते हैं, sucrose को invert sugar क्यों बोलते हैं, we'll see in the upcoming slide. ऊपर क्वेश्चन भी ऑलरेडी आ चुका है. Why sucrose is called as a invert sugar? So see in upcoming slides. Before that, disaccharides, the first example of disaccharide is a sucrose. Second example is a lactose. Lactose is known as a milk sugar. ये मिल्क में पाया जाता है. Lactose is known as a milk sugar. Lactose is known as a, because it is found in the milk. Now lactose is a disaccharide. Di मतलब क्या, 2, मोनो मतलब 1, डाई मतलब 2, ट्राई मतलब 3, टेट्रा मतलब 4, पेंटा, हेक्सा, हेप्टा, octa, nona, deca. On the basis of that, monosaccharide means only single sugar unit, glucose, fructose, mannose, है ना, राइबोज, एक सिंगल यूनिट है. डाइसैकराइड मतलब क्या, 2 मोनोमर से. जैसे सुक्रोज में क्या है, ग्लूकोज प्लस फ्रुक्टोज. जैसे लैक्टोज में क्या है, तो इन द लैक्टोज यू विल फाइंड ग्लूकोज प्लस गैलेक्टोज, ग्लूकोज प्लस गैलेक्टोज, ग्लूकोज प्लस गैलेक्टोज. व्हाट इज द ग्लाइकोसिडिक बॉन्ड, यहाँ पे ग्लाइकोसिडिक बॉन्ड कौन सा है, बीटा डी गैलेक्टोसिल 1,4 बीटा डी ग्लूकोज. मतलब क्या, ये गैलेक्टोज मॉलिक्यूल जो है उसका 1 कार्बन, फर्स्ट कार्बन एंड 4th कार्बन ऑफ ग्लूकोज, है ना. गैलेक्टोज का फर्स्ट कार्बन मतलब फंक्शनल कार्बन, इसका जो फंक्शनल कार्बन है वो, और इसका फोर्थ कार्बन मतलब नॉन फंक्शनल कार्बन। सो इसमें एक एल्डिहाइड फ्री है। सो इन द लैक्टोज मॉलिक्यूल the aldehyde group of this glucose is free, so that's why it is a reducing sugar. The reducing sugar. But if you see the structure of fructose, alpha D glucosyl, first carbon is a functional carbon, and 1 2 beta D fructose. Fructose का functional carbon कौन सा है 2. So both the aldehyde group of glucose and ketone group of fructose both are involved in the glycosidic bond formation. That's why this sucrose is a non-reducing sugar. Non-reducing. Third है ना, lactose is known as a milk sugar. Remember, that is the maltose. Maltose मतलब क्या है? Maltose, in the maltose glucose plus glucose. Glucose plus glucose, two molecules of glucose, they are combined by a glycosidic bond in the maltose. Linkage क्या है? तो beta D glucosyl 1,4 beta D. Also known as a malt sugar. Maltose is known as a malt sugar. Maltose को बोलते हैं malt sugar. It is a hydrolysis product found in some germinating seeds. जो सीड्स जो होते हैं, जैसे हम है ना सुबह सुबह आप स्प्राउट खाते हो, स्प्राउटिंग जो होता है, इन द स्प्राउट्स द शुगर फाउंड इज अ maltose. In the sprout, sugar found in the maltose, because in the germinating seeds you will find this sugar. You can see. So that's why maltose on a hydrolysis will give you 2 molecules of D glucose. Maltose after hydrolysis will give you 2 molecules of glucose. Lactose on hydrolysis will give you 1 D galactose and 1 D glucose. Sucrose on hydrolysis will give you D glucose and D fructose. तो इसके जो disaccharides है, on the linkage of disaccharides, lot of questions has been already there in a GPAT Niper DI Pharmacist. So linkage is important, this linkage is important, okay, and then synonyms are important, reducing, non-reducing, that part is important. Multiple times question was asked in the exam. You can see the structures of these sugars. Okay. So this is the structure of sucrose. This is the structure of, this is a glucose component and this is a fructose component. So 1 2. So 1 मतलब क्या, aldehyde containing group, functional group of glucose. This is involved in the bond with the functional group of fructose, that is ketone. That's why it is a non-reducing sugar. Non-reducing. क्या है, sucrose is a non-reducing. Now if you see the structure of maltose, maltose क्या है, glucose plus glucose in a 1,4 linkage. First carbon atom of one glucose molecule forms a glycosidic bond with the fourth carbon atom of another glucose molecule. So from this glucose molecule, aldehyde is engaged in the bond formation, but the second glucose molecule has a free aldehyde. Second glucose molecule has a free aldehyde group. That's why it is a reducing sugar. So maltose is a reducing sugar. Now lactose. Lactose is a 2 component, galactose and glucose. Galactose and glucose means lactose. Galactose and glucose means lactose. Now first carbon atom of glucose, that is a functional carbon involved in the bond formation with the fourth carbon of galactose. Galactose का first carbon मतलब functional carbon forms a bond with the fourth carbon of glucose, but in the second molecule जो second glucose है उसमें का aldehyde free है. The aldehyde group of second molecule glucose is free, so that's why galactose is also reducing sugar. What is the definition of reducing sugar? Reducing sugar means what? Sugar having free aldehyde or ketone group. अगर फ्री अल्डिहाइड और कीटोन ग्रुप है शुगर में तो उसको बोलेंगे अपन रिड्यूसिंग शुगर। तो देखो इसमें ये डाइसैकेराइड है, लेकिन एक एक ग्लूकोज का मॉलिक्यूल है ना, एक तो फ्री है, सो देयर इज वन फ्री अल्डिहाइड ग्रुप रिड्यूसिंग शुगर। हियर आल्सो देयर इज अ one free aldehyde group to reduce sugar. But if you see the example of sucrose में no free aldehyde or ketone group, because ketone group of fructose and aldehyde group of glucose both are involved in the bond formation, both are engaged in the bond formation. That's why sucrose is a non-reducing sucrose is a non-reducing sugar. I hope you have understood. देखो disaccharides. So in the disaccharide, what is the formula? General formula is nothing but the C12H22O11. C12H22O11. That is the formula for disaccharide. Remember C12H22O11. When two monosaccharides are joined or linked together by a glycosidic bond. When two monosaccharides are linked together by a glycosidic bond or joined together by a glycosidic bond, so that form a disaccharide. Properties of disaccharide depends on the type of linkage present. So examples of disaccharide कौन सा है, अपने ऑलरेडी देखा है, सुक्रोज, माल्टोज, लैक्टोज, दे आर इंपॉर्टेंट डाइसैकेराइड। सुक्रोज जो है मेड फ्रॉम ग्लूकोज एंड फ्रुक्टोज इज नोन एज अ इनवर्ट शुगर। सुक्रोज इज आल्सो नोन एज अ इनवर्ट शुगर। इसके ऊपर क्वेश्चन 2-4 बार आ चुका है। व्हाई इट इज नोन एज अ इनवर्ट शुगर? सी, सुक्रोज इज इनवर्ट शुगर, इट इज अ नॉन रिड्यूसिंग and does not forms oxazone. So properties of sucrose is table sugar, cane sugar or invert sugar, it is a non-reducing and does not form a oxazone. Both functional group, that is aldehyde and ketone, are involved in the bond formation. Maltose क्या है? Maltose and lactose forms a oxazone because functional group is free, one of the participating monosaccharide, है ना। तो पहला देखो maltose, पहला डाइसैकेराइड कौन सा है? माल्टोज, also known as a malt sugar. It is obtained by hydrolysis of starch. स्टार्च का अगर हाइड्रोलिसिस करेंगे तो माल्टोज मिलता है। इट इज अ स्वीट इन टेस्ट, सोल्यूबल इन द वाटर, रिड्यूसिंग शुगर है। इट गिव्स अ सनफ्लावर शेप्ड ओसेजोन्स। इट इज डाइजेस्टेड बाय अमाइलेज एंड माल्टेज, है ना। डाइजेशन होकर क्या बनेगा? माल्टोज इज कंपोज ऑफ 2 मॉलिक्यूल्स ऑफ ग्लूकोज। Now lactose क्या होता है? It is a milk sugar. Lactose is known as a milk sugar up to 5% found in the milk, found as a equilibrium mixture of alpha, beta and beta. Alpha D lactose and beta D lactose होता है 2:3. It is a very water soluble and not sweet in taste. The dextrorotary, also it is a reducing sugar and forms a powder puff shaped or hedgehog shaped osezones. Now in the GIT of children's, जो बच्चे होते दूध कौन पीते हैं? बच्चे पीते हैं, बाकी सभी पीते हैं आजकल। But there is a enzyme known as a lactase in the intestinal juice. There is an enzyme known as a lactase in the intestinal juice. GIT है, उसमें lactase enzyme होता है that causes the hydrolysis of lactose. There is a maltase enzyme that hydrolyzes the maltose. There is a sucrase enzyme that hydrolyzes the sucrose. There is a one test known as a Heron's test, which is used to distinguish between lactose and sucrose and other monosaccharide. Remember ये टेस्ट का नाम Heron's test. Heron's test is used to distinguish between lactose, sucrose and other monosaccharide. Okay, in the few people देखो there is one disease known as a lactose intolerance, है ना। जो ऐसे डिसीज होते हैं जो बड़े लोगों को होते हैं, स्पेशली फॉर एक्टर्स एंड ऑल दैट, तो फ्यू पीपल दे कैन नॉट टॉलरेट द मिल्क, अगर उन्होंने मिल्क पी लिया तो उनको डायरिया हो जाता है। अगर बहुत सारे लोग होते हैं जिन्होंने अगर दूध पिया तो उनको डायरिया होता है। क्यों होता है? बिकॉज दोज आर द सब्जेक्ट्स they are not having lactase enzyme in their GIT, that's why not able to break down the lactose, that's why lactose causes the diarrhea in those kind of subjects। तो कुछ कुछ लोगों में दूध पीने से क्या होता है? डायरिया होता है। क्यों होता है? बिकॉज दे आर नॉट एबल टू डाइजेस्ट द lactose because of the deficiency of lactase enzyme, especially for example Priyanka Chopra, you might know, the Bollywood actress, so she is also having this lactose intolerance। And Pierron's test is used to distinguish between lactose, sucrose and other monosaccharide। Now sucrose क्या है? Sucrose is also known as cane sugar or table sugar or invert sugar। के ऊपर multiple times question। So what is sucrose? Obtained from the sugar cane or sugar beets or sugar apples or sugar apples, also found in the fruits and vegetables। बहुत सारे fruits में भी आता है, vegetables में आता है। Very soluble in the water and very sweet in taste ना। It is a dextro rotary। इसका ऑप्टिकल रोटेशन क्या है? सुक्रोज का डेक्स्ट्रो। सुक्रोज इज अ डेक्स्ट्रो rotary, non reducing, because both aldehyde and ketone of glucose and fructose are involved in the bond formation, right? Non reducing and does not show muta rotation, does not form a oxidation formation। Now sucrase is the enzyme in the GIT that hydrolyzes the glucose forming glucose plus fructose। Now why sucrose is known as the invert sugar? ऊपर मल्टीपल टाइम्स क्वेश्चन आ चुका है ना, inversion of sugar। Why sucrose is known as the invert sugar? The answer of this question lies in this paragraph। Inversion of sucrose, or why sucrose is called as the invert sugar? The answer of this question lies in this paragraph। You see, if you check the optical rotation of freshly prepared sucrose solution, अगर आपने सुक्रोज का सोल्यूशन प्रिपेयर किया, उसका ऑप्टिकल रोटेशन चेक किया, तो इनिशियली विल कम डेक्स्ट्रो, इनिशियली वो डेक्स्ट्रो रोटेशन दिखाएगा। सुक्रोज इज अ डेक्स्ट्रो रोटेटरी, है ना। एंड नाउ इफ यू ऐड सम एसिड आफ्टर हाइड्रोलिसिस, if you add some acid in sucrose solution, so hydrolysis product of sucrose क्या होगा? That is a glucose and fructose। Glucose is dextro, fructose is LiO, but total hydrolytic product of sucrose is a LiO rotary, because fructose has a greater specific LiO rotation than the dextro rotation of glucose। मतलब जब हाइड्रोलिसिस होगा तो ग्लूकोज तो डेक्स्ट्रो है, फ्रुक्टोज तो LiO है, but dextro rotation of glucose is overcome by LiO rotation of fructose, and that's why final mixture of hydrolysis is having LiO rotation। Means original fresh solution of sucrose was a dextro, but after hydrolysis it become a LiO, that means the rotation is inverted, that's why sucrose is known as an invert sugar। that's why sucrose is known as an invert sugar। तो देखो फ्रेशली अगर सुक्रोज का सॉल्यूशन बनाएंगे, है ना, सुक्रोज का सॉल्यूशन, तो उसका अगर आप ऑप्टिकल रोटेशन चेक करेंगे वो डेक्स्ट्रो आएगा। उसमें अगर आपने एसिड ऐड कर दिया, एसिड, तो क्या होगा? हाइड्रोलिसिस होगा, है ना। हाइड्रोलिसिस होने के बाद में हाइड्रोलिसिस प्रोडक्ट जो उसका हाइड्रोलिसिस का सॉल्यूशन जो है उसका अगर ऑप्टिकल रोटेशन चेक करेंगे, दैट विल कम अ LiO, है ना। ओरिजिनल वाज अ डेक्स्ट्रो and finally is a LiO, that's why it is known as an invert sugar, and the process of this is known as a inversion। honey is the largely known as the invert sugar। जो हनी होता है, जो आपका हनी खाने का बोल रहा हूँ मैं, आपका जो हनी है वो नहीं, आपका खाने का जो हनी होता है दैट इज नोन एज द इनवर्ट शुगर ना। is more because it has a more presence of fructose। Now lactulose क्या है? lactulose is a keto disaccharide। keto disaccharide obtained by heating the milk। जब मिल्क को हीट करते हैं तब लैक्टोज बनता है उसमें. Commercially nowadays it is synthesized synthetically. It is not digested in the intestine but fermented by intestinal bacteria and used as osmotic laxative. लैक्टोलोज जो है वो लैक्सेटिव है, लैक्सेटिव भी है. ब्रांड नेम ऑफ लैक्टोलोज डुपलैक. डुपलैक नाम का एक डुपलैक नाम का टैबलेट भी आता है, सिरप भी आता है. एबॉट फार्मास्यूटिकल. That is a lactulose, which is a osmotic and an osmotic laxative. So remaining points will take tomorrow. Tomorrow we'll see the polysaccharides. Polysaccharide देखेंगे, and after that we'll see polysaccharides के बाद में देखेंगे heteropolysaccharide वगैरह, है ना. And after that we'll discuss the utilization of glucose in the body. Then we'll see the metabolism of glucose. मेटाबॉलिज्म देखेंगे ग्लूकोज का, है ना. बाद में ये मेटाबॉलिज्म खत्म होने के बाद में एंटी डायबिटिक ड्रग्स विल स्टार्ट, so that you will easily understand. I hope you have understood today's session. See you in